Hello, and welcome to our webinar. I'm Jason Winnike, Senior Study Director here at Metabolon. Today, we'll be talking about how some of our new targeted quantitative panels can help advance your inflammation-related research. Here's a look at our agenda for today's presentation. We'll first talk about inflammation in general, then we'll move on to the benefits of integrating metabolomics into your inflammation-focused research, touching on relative versus absolute quantitation. Then we'll talk about 4 new quantitative targeted panels that we've developed here at Metabolon, which can be used in inflammation research. Inflammation is a normal component of the body's immune system and is initiated to defend against acute insults, such as those involving pathogens and infections. When these pathways work correctly, they are very beneficial to health, but inflammation can also be a driving force behind disease. The importance of inflammation in developing multiple diseases and health-related conditions, including neurodegeneration, diabetes, cardiovascular disease, cancer, and inflammatory intestinal diseases, is undisputed. Inflammation is an immunological defense response to harmful stimuli, such as physical trauma or infection, which begins when activated leukocytes migrate to the affected area and release various short-lived pro-inflammatory substances, such as cytokines, prostaglandins, and histamine. This protective response, characterized by redness, swelling, heat, and pain, helps the body to clear the pathogen or injury. Then repair when the pro-inflammatory substances dissipate, shifting to an anti-inflammatory signals which resolve inflammation. Thus, inflammation occurs to resolve the injury or irritation and initiate the healing process. As such, it can be beneficial. Prolonged or chronic inflammatory states, however, are associated with negative health consequences. So inflammation is a normal response to a threat, but certain factors such as social, psychological, environmental, and biological factors can prevent acute inflammation from resolving, leading to chronic low-level inflammation. Chronic inflammation has been linked to cardiovascular disease, diabetes, chronic kidney disease, non-alcoholic fatty liver disease, autoimmune disorders, neurodegenerative disorders, and cancer. Greater than 50% of all deaths, in fact, are attributable to inflammation-related diseases. So how can metabolomics help with inflammation-related research? Well, metabolomics can inform on inflammatory processes by providing a readout of the small molecules of an organism, tissue, biofluid, etc. This allows for direct molecular phenotyping of the small molecules directly involved in promoting and resolving inflammation, beyond that which is measured in other assays that measure macromolecules such as proteins. Both global and targeted metabolomics have many great applications for inflammation research. Metabolomics can be used for the identification of biomarkers. It can be used to investigate the effects of treatments for various inflammatory diseases. It can be used to investigate the relationship between inflammation and related diseases. Additionally, the metabolic pathways affected by inflammatory diseases can be investigated using metabolomics. Metabolomic profiling can be conducted globally by assessing many metabolites and pathways, or by using a targeted approach. By using one of Metabolon's quantitative targeted panels, you can take a targeted deep dive into specific metabolic pathways and classes of molecules. In doing this, many benefits are gained, such as absolute quantification. Let's dive deeper into quantification. For global metabolomics analyses, it is not currently possible, in a practical sense, to analyze the 5,000 plus metabolites in the library and obtain absolute concentrations for all analytes. However, when targeting a limited number of predefined metabolites, as is done in our targeted panels, standard curves that contain the metabolites of interest are analyzed at different concentrations, which, along with labeled internal standards, allow us to calculate the concentrations of the metabolites using quantitative units, nanograms per milliliter, for instance. When the metabolites are unitless, like in a global analysis, comparisons can be made between samples or between groups for a given metabolite. So you could say cholesterol doubled when going from time point 1 to time point 2, but you could not say that at time point 2, there was twice as much lactate as there was glucose, since the measurements are unitless. With absolute quantitation, you can make these comparisons. You can even calculate ratios between different metabolites, which can be informative. With a global analysis, sample processing and analytical methods are chosen that are generally good for a wide range of metabolites: polar, nonpolar, large, small, concentrated, and dilute, and everywhere in between. With a targeted panel, sample processing and analytical methods are focused on optimizing the analysis of a limited number of metabolites, generally chemically similar metabolites. The consequence of this is greatly enhanced sensitivity. Low concentration metabolites, or metabolites that otherwise are not well detected in a global analysis, can be well detected in a quantitative targeted panel due to this method optimization. At Metabolon, we recognize the value of targeted metabolomics and absolute quantification to help our customers further their research. This slide showcases our suite of targeted panels that provide absolute quantification of metabolites related to inflammatory pathways. This webinar will focus on the newly developed panels showcased at the top of the slide: the Oxysterols, Sphingolipids, lipid mediators of inflammation, and Cannabinoids targeted panels. The first panel I would like to describe is Metabolon's Lipid Mediators of Inflammation targeted panel. Lipid mediators of inflammation are bioactive lipids that can have pro-inflammatory activities, which act at the beginning of the inflammation process, or anti-inflammatory activities that function at the end of the inflammation process. This targeted panel measures lipid species from 6 metabolite classes, from the fatty acid precursors, such as arachidonate, to oxidized fatty acid intermediates and bioactive lipid mediators with well-established pro- and anti-inflammatory properties. Our validated method for lipid mediator quantification can enable you to gain insights into the production of bioactive lipids within your cohorts. It's a large panel with 58 metabolites spanning multiple biochemical classes. It has been designed and validated for use with human plasma samples, and it features a 6 to 8 week turnaround time. When thinking about inflammation and small molecules that are associated with inflammation, we typically think about pro-inflammatory eicosanoids. There are multiple subclasses of pro- and anti-inflammatory lipid mediators. To initiate inflammation due to an infection, for instance, polyunsaturated fatty acids, such as arachidonic acid, are released from cell membrane phospholipids. These polyunsaturated fatty acids are the precursors to pro-inflammatory eicosanoids, such as leukotrienes, prostaglandins, and thromboxanes. To resolve the inflammatory process, anti-inflammatory lipid mediators, such as lipoxins, resolvins, protectins, and myrisins, are often produced in response to pro-inflammatory signaling molecules and work to counteract the effects of the pro-inflammatory molecules by inhibiting their production or activating anti-inflammatory pathways. Generally speaking, omega-3 polyunsaturated fatty acids often serve as substrates for anti-inflammatory lipid mediators, while omega-6 polyunsaturated fatty acid function as substrates for predominantly pro-inflammatory mediators. Consequently, omega-3 and omega-6 fatty acids, as well as the resulting lipid mediators, can be influenced by the profile of polyunsaturated fatty acids ingested in the diet. As you can imagine, the lipid mediators of inflammation panel has wide applicability and can be leveraged for any study where inflammation is a phenotype of interest. This would include, for example, studies involving injuries and wound healing, infection, health and wellness, stress, chronic inflammation, and diseases associated with chronic inflammation, such as neurodegenerative diseases, atherosclerosis, diabetes, asthma, autoimmune disorders, inflammatory bowel diseases, periodontal diseases, and cancer. The second panel I would like to introduce you to is the Oxysterols targeted panel. It supports neuroscience, cardiometabolic disease, cancer, and diabetes research. Oxysterols are oxidized derivatives of cholesterol that serve as precursors to other metabolites, such as bile acids. Of interest, in the context of inflammation, many oxysterols are also bioactive and can be used as disease biomarkers. Metabolon's oxysterol panel measures cholesterol and 11 other cholesterol precursors and oxidized products of cholesterol. Our validated method for oxysterol quantification in human plasma samples can enable you to gain insights into the biological functions of this important metabolite class within your cohorts. Oxysterols are typically present at extremely low concentrations, especially compared to cholesterol, from which oxysterols are derived. which is present at a high concentration. The large concentration range of the metabolites in the panel and the fact that oxysterols can be produced during sample processing through oxidation make detection of these metabolites analytically challenging and technically demanding. Fortunately, we've already done the heavy lifting in creating and validating this panel. There are 12 metabolites targeted in this panel, and it is designed for use with human plasma samples. It features a 6 to 8 week turnaround time. Cholesterol can be relatively easily oxidized, transforming it into various oxidation products known as oxysterols. Oxysterols can be formed non-enzymatically, such as from reactive oxygen species, which makes oxysterols a biomarker for oxidative stress, or they can be formed enzymatically, such as from cytochrome P450. Enzymatic oxysterol production is important for generating precursors for certain metabolites, such as vitamin D and bile acids. The brain must synthesize all its cholesterol because the blood-brain barrier prevents cholesterol from passing from the blood to the brain. The central nervous system contains about a quarter of the cholesterol present in the body, which is the highest content of any tissue. This high level of cholesterol means that oxysterols can be used as biomarkers for neurodegeneration. Oxysterol equilibrium in human body fluids can be disturbed by some diseases. Therefore, some oxysterols can be used as biomarkers for disease diagnosis and monitoring. While we have a large supply of dietary cholesterol, most cholesterol, about 70%, is synthesized in the cells. Therefore, oxysterols can be used as biomarkers of altered cholesterol metabolism or disorders involving cholesterol synthesis. Many oxysterols are bioactive molecules. De novo cholesterol metabolism is also regulated, in part, by oxysterol levels. Some inflammatory and immune responses are influenced by the presence of oxysterols. They have also been shown to induce apoptosis and necrosis. In fact, certain diseases are thought to be caused or exacerbated by pro-inflammatory, pro-apoptotic oxysterols created by high levels of cholesterol or perturbed cholesterol metabolism. For example, oxysterols have been shown to influence the initiation and progression of atherosclerotic plaques. Additionally, oxysterols are known to regulate certain signaling pathways that are required for proper cell differentiation during embryogenesis. Taken together, oxysterols should be regarded as an important biological class, and Metabolon's oxysterol panel can help you elucidate their function in these various processes. The next panel I'm going to highlight is Metabolon's Sphingolipids targeted panel. It can support a large range of research topics associated with inflammation, including cardiometabolic disease, neurological disease, and cancer research. Sphingolipids are a class of lipids with a sphingoid base and the fatty acid amide bonded to the amino group at the C2 position. The 5 classes of sphingolipids measured on Metabolon's Sphingolipids targeted panel include dihydroceramides, ceramides, hexosyl ceramides, lactosyl ceramides, and sphingomyelin. While variations are possible within the sphingolipid base structure, this panel specifically measures dihydroceramides and ceramide species, sphingonine and sphingosine bases, which correspond to the major forms found in mammalian systems. There are 61 metabolites in the panel. It is designed and validated for use with mammalian serum or plasma samples and features an 8 to 12 week turnaround time. Sphingolipids are structurally unique lipids that play roles in membrane integrity and fluidity. However, many sphingolipid species are bioactive molecules and regulate multiple biological processes, such as cell growth, proliferation, migration, and apoptosis. Consequently, their cell signaling properties have been implicated in a number of diseases of inflammatory origin, including cardiovascular disease, neurodegenerative disease, cancer, and diabetes. In this context, much research focus has been on ceramide, which is positioned as a central hub in the sphingolipid metabolism pathway, as shown here. Of particular note, circulatory sphingolipids have been found to change dynamically in response to tissue injuries, which has led to their identification as predictive biomarkers for various cardiovascular disease outcomes. Ceramide synthase, the enzyme that catalyzes the de novo formation of dihydroceramide and ceramide, has 6 major isoforms that differ in regard to their tissue distribution and specificity for chain length of the acyl-CoA donors. Thus, the acyl chain length in ceramides can, in part, inform the location of origin. Having the granularity of the distinct sphingolipid species offered in this targeted panel, for instance, being able to specifically measure ceramide 16-0, which has been shown to have pro-apoptotic signaling properties, allows for a deeper dive to be made in sphingolipids analysis. With the targeted panel being quantitative, comparisons between different specific sphingolipid species can be made, which, as noted, can expand the possibilities for biomarker identification. Ceramides and hexosyl ceramides of longer chain lengths have been shown to accumulate in Alzheimer's disease patients, for instance. And through examination of individual sphingolipid species, these kinds of discoveries can be made. The interconversion between ceramide and sphingomyelin is also of key importance, particularly in the context of inflammation. Sphingomyelinase enzymes, which catalyze a conversion of sphingomyelin to ceramide, are known to be activated by pro-inflammatory mediators, including TNF-alpha. Additionally, ceramide can activate inflammatory pathways through upregulation of transcription factors such as NF-kappa B, and their levels have been found to correlate with inflammatory cytokine production. Thus, being able to measure these sphingolipid species with different acyl chain lengths and being able to make direct quantitative comparisons within a given class and between different classes can help one better understand disease pathogenesis. The final panel I would like to discuss is Metabolon's Cannabinoids targeted panel. It supports immunology, neuroscience, and oncology research. Cannabinoids consist of plant-derived phytocannabinoids, which are multi-ring phenolic compounds, and endocannabinoids, which are endogenous lipid-based metabolites. The three classes of cannabinoids measured on Metabolon's Cannabinoids targeted panel include cannabinoids and related molecules, phytocannabinoids, and human metabolites of phytocannabinoids. There are 31 compounds in the panel. It is designed and validated for use with human plasma samples, and it features a 6 to 8 week turnaround time. Cannabinoid signaling pathways are essential for maintaining normal neurological functions, including memory, locomotion, appetite, and pain, and research indicates that cannabinoids play a role in the maintenance of neuronal health and the prevention of neurodegeneration. Ongoing studies are investigating the use of cannabinoids in the treatment of Parkinson's, Alzheimer's, Huntington's, and epilepsy. Due to their anti-inflammatory effects, cannabinoids have been proposed for treating immune-related diseases such as rheumatoid arthritis, colitis, multiple sclerosis, and some liver diseases. While cannabinoids have received FDA approval for treating nausea caused by chemotherapy, animal models have shown that cannabinoids can slow and/or inhibit cancer growth. Studies have shown cannabis to be effective in treating chronic pain, offering an alternative to opioids. And finally, this panel could be used for monitoring clinical study subject compliance with respect to cannabis usage for inclusion, exclusion, or washout period criteria. The targeted panels we have talked about today are available from us as fee-for-service metabolomic solutions. Fee-for-service metabolomics allows you to leverage our industry-leading methods and deep scientific expertise and can be more cost-effective than doing metabolomics in-house. You can easily scale up or scale down your projects as needed, providing flexibility in the management of projects and timelines. We offer discovery panels with relative quantification and targeted panels and assays with absolute quantification, such as the ones I've talked about here today. We operate using procedures established in our quality management system to ensure accurate and consistent deliverables. We employ robust quality assurance and quality controls at every stage, with a rigorous data governance program and adherence to valued U.S. federal and international standards and regulations. Through Metabolon's end-to-end service for all our products, from study design to data reporting, we strive to provide maximum value, utility, and applicability of the data that we generate to advance your research objectives. We work with you every step of the way to make sure your samples are prepared, shipped, and processed to ensure the highest quality data possible, so you can identify meaningful biological insights. Our unparalleled experience and extensive expertise make Metabolon the ideal metabolomics partner. Thank you for joining our presentation today. Feel free to reach out via our website if you have any questions or want to connect with a metabolomics expert.